Okay, we are doing chapter 14. If you did miss chapters 10, 11, 12, and 13, which is a lot of you guys, unless you're rewatching this, this.
If you're finishing with nothing,
What?
nothing.
So if you did for those 4 chapters, please, I think Matt could do that again, those ones again. Giselle, maybe you should, bug Matt to do those ones.
Which ones?
The 10, 11, 12, and 13.
For what? For, like, review? Or?
Yeah. Yeah.
So when you start the study sessions to go over those chapters in particular?
Yeah.
Okay, sure.
Because I didn't record it because I thought it was pretty easy. Steven thought it was pretty easy. Noah grasped it really well, and I just forgot to be honest.
It's okay, you're not obligated.
I know. Okay, let's talk about DNA. DNA is interesting. Because this is where the hardest stuff is. This is how we learned how DNA is hetery, which means it can be passed down because it can survive heat. See, here, it can survive because it can be passed down. DNA can be transported while proteins cannot because they can denature from heat. DNA is very resistant, more resistant than proteins. Okay. This guy, he discovered adenine and thymine are the same, and guanine and cytosine are the same. I don't think you need to know that. Double helix, which means the rotating, it's like the double spiral covalent bond.
There's no lesson for this one that they discovered the double helix.
Yeah, they just discovered this is just really what it is that it's heredity because it can survive high materials. This we've discussed this in Steven, we discussed this in chapter 10, 12 or something, or
yeah, I think it was 12.
Yeah, with genetics. No, not even genetic. I think it was mitosis. But it goes nucleotides, then a series of nucleotides make a gene. A gene is a group of nucleotides that code for something. Then a histone rolls that up into a nucleosome, and nucleosomes form chromosomes. That's how it goes. DNA, this is like an exact slide. Really, you got hydrogen bonds between thymine and adenine, and cytosine and guanine, and they're bonded. Then you got phosphate, nitrogen groups, all these stuff. These ones we've talked about already. Giselle, if you, I just want to say, please bug Matt for those ones. Because these are because these are repeats. This slide is an exact repeat, right, Steven? I've seen this slide. So now we're going to deal with DNA replication. This is where the hard part is. I don't want to waste my time on this. So DNA replication is, I think every picture we do, it gets more complex and complex. So, yeah, every picture we do, it gets more complex and complex. Okay. So let's start with the most simplest of pictures. So, can you describe what is happening to me? Just describe what you see in the picture. Just forget about the words, just describe what's in the picture.
The enzyme is opening up the helix, and then the primer is coming, and the DNA polymerase makes the complementary strand, and the DNA ligand seals it up.
Yeah, exactly. Basically, enzymes come in, break the helix, primer comes in, and primer isn't filling things in. It's just like a base. It's a base. So the DNA polymers will come in and put the nucleotides, the complementary base, which means, let's say, if I have guanine, then the complementary base of that would be cytosine. And then DNA ligase, what it does is it closes any holes left, and the double helix is reformed. Let's start with our second picture. What is this one telling you?
This one adds in the replication fork, right?
Yeah.
So yeah, it's kind of like adding on to the last picture.
Like more in depth.
Yeah.
Yeah, this one, maybe it's labeled more in depth, but I like the first picture actually better. In my opinion, this picture looks like tells more of a story than this one. It's really, you cut it in half and then put the things in. So I don't know if this really is.
It just looks a little bit more complicated than the second picture.
Yeah, but this one I think gives more information in a cleaner way.
Yes, that's true.
Let's look at this one. So, adding nucleotides.
Yeah. It's showing you the phosphate and the hydrolysis so that you could add the nucleotides.
Hydrolysis. Oh, so for the energy for this, we don't need ATP for this because we make our own energy by breaking the phosphate in the edges. The edges of DNA and RNA is made out of phosphate. So by breaking it, we get energy. Okay, this picture is the one I think this is the picture we went most in class over.
Yeah, it's gonna be this one.
So we have two strands. We have the lagging strand and we have the leading strand. So the leading strand will go over here, and the enzyme unwinds. One will be the leading strand, and this strand is nice and clean. DNA polymers will just easily come in and fix it. But the second one we have is the lagging strand. This one isn't so easy. We have Okazami fragments, we have the RNA primer, we have DNA polymers, we have all these things. So in this lagging strand, we're going to use RNA primer because we have to wait. We have to wait for this to unwind first for this to arrive. We need to wait until the leading strand unwinds for the other one to unwind. So what will happen is this RNA primase will add primer will add primase, but it'll jump some. It'll jump some areas, it'll jump as more gets unwinded. Then you got your DNA polymers. What the DNA polymers, it digests or it removes the primer and adds nucleotides. But what happens is you're left with an Okazami fragment or holes. You're left with the holes in it, which the DNA ligase will cover up. Is that pretty much it? The bubble. I'm kind of confused about the bubble.
No, this is just another way of showing the thing. Like, this is just, I don't know why it's in a bubble form, but this is just the same picture, just a different way of showing the same process.
I hope.
Yeah, it's not, these are all, this is the emphasis on the lagging strand. The other one's the emphasis on the leading strand.
Yeah, because the leading strand is just clean, continuous.
That goes like, but the other one is just going back and forth.
Yeah, lagging, back, forth, and then you get Okazami fragments that DNA ligase fixes. Yeah. So really, this one is I like this picture better. I don't know why. I don't like the green blobs. The green blobs really hurt the picture.
It's a nice video. I can link. It's also in this slide for like replication.
Which one?
And it goes over it really well.
Is it the Aniva sisters or the Few School?
I don't know if we had looked up a video or but yeah, it showed the leading and lagging strands.
Yeah.
The replication, and it was really simple.
Yeah, if you guys have time, One thing, because I'm not admin, I want like a chat, like a general chat where we can put all our links. All talking should be like deleted. Anything other than links should be deleted on that.
Wait.
Cause look, I mean,
Yeah, we gotta talk to Matt.
yeah, cause all the links and stuff like that get like they always go up, so you have to scroll up for them. Like my poster for chapter 4, the videos for 4, 5, 6.
Yeah, the link, like you can't pin everything too.
Yeah, so
like a channel that's specifically linked only.
that would have been assignments, but then we started talking in assignments. Like there would be like some sort of conversation that happens at assignments, announcements. So that's not really a good place to do it. I think it should be like, yeah, just links. If I see anything other than links, if I see anything other than links, it should be just deleted. Okay, so really this picture is what's showing what's this picture. I forgot. Does it go 3, 5?
It's gonna go
3 to 5?
To the 5, I believe. Let me look.
I think it says in the third picture, the one with the green
5 to 3. 5 to 3.
Yeah, oh, okay.
It goes from a 5 to 3. 5 to 3. Oh, this is why this is the lagging strand. This is the leading strand, because look, it starts over here and then moves like this. This one moves 5 to 3. This one moves this way, the same way. I'm just confused about direction. Just it moves 5 to 3. So once we do that, we have to repair. Name something that can harm our DNA.
Denaturation.
Yeah.
Like high temperatures, I guess.
High temperatures, UV light, gamma rays, X-rays. Things like that. Or replication errors can occur. Maybe the DNA ligase doesn't do everything. Maybe there's a mutation in the entire process. So A might change to C. So DNA polymers works quickly. That's the problem. It works quickly. Damaged or broken DNA. It could miss damaged DNA. It could miss broken DNA. There could be a loss of nucleotides or extra nucleotides. We'll talk about mutations. But once you lose and gain nucleotides, it becomes a mutation. It can be proofread, and DNA repair enzymes are there. They'll be there to fix any changes. But there are mutations that occur. Mutations are permanent changes in DNA sequence when proofreading and repair mechanisms fail. So this fails. If there's a problem here, this fails. All this fails, we get a mutation. So you got UV rays can harm it. Gamma rays, anything above 320 nanometers in length. Chemicals. So let's talk about types of mutations. What is substitution?
Substitution is where you take one of the nucleotides and you put a different one that's not supposed to be
You're
there.
substituting, you're replacing. It's like plugging in math. When you substitute something in math, you're plugging something in. Substitution. Insertion. What is insertion? And what makes this special? This
that's not supposed to be there.
What makes it special? Or what makes it worse than substitution?
Because substitution, it can be corrected, but if you insert something, it throws off the entire sequence.
So basically anything that is after the thing that got mutated changes. Because you have to move everything to the one. To the side. Because nucleotides amino acids can only be created in 3s. That's a codon. So when you're adding a nucleotide, you're changing the entire codon from that codon to the next one to the one after that. And what about deletion?
It's the same thing but reversed. Instead of inserting, you delete one and it throws off the sequence.
Point mutations that alter codons. What is a codon? Codon is 3 nucleotides that code for a proton, amino acid, not protein, amino acid. So you got silent. What is silent? It has no change. You got, what's B? Minence. Hello, Howard. We have minence. I think misnence. I don't know how to say that. So the thing is...
What? Which one?
B. Misnence.
Where? Point to it. Use your cruiser. Oh, okay. Oh my gosh.
And
Missense.
Minsense. And
No, like myth and then sense.
Minsense. And this one you're making a stop codon. This one.
I mean, I don't think we have to know like this one specifically.
Oh, that's slightly different amino acid sequence. This one is slightly. Nonsense is you just make a stop codon. Frame shift insertion is insertion and deletion. Provide an example / disorder caused by a mutation. What would be one disease?
I think we talked about sickle cell anemia for this one.
Yeah, and how hemoglobin is altered. Proofreading. E. coli mutates with DNA synthesis error rates 100 times higher than normal. Why is that? Why does E. coli and other bacteria mutate 100 times faster than us?
Because, that's a good question, because I literally forgot.
It's because they like mutations. Because they're like so little and so used, like billions of them. There's
gonna say to gain the antibiotic resistance.
Yeah. They benefit from mutations because something bad happened, I guess you died, but there's billions of you. And if something good happens, you can spread it. Adds a mispage. So here is what proofreading does. Someone will proofread it, see something's wrong, and then replace it. And this happens in DNA polymers, also proofreads. Mistakes happen in 1 in 10 million bases. So they're pretty rare, but they're still possible, so proofreading will help you. So let's say UV light hits you and your DNA became funky. So you have a mechanism of repair. I think there's a picture for this. Yeah, like right here. I think like this picture. This is a good mechanism of repair. So UV light hits or chemicals, and it can change. It can move this and bend it. So the thymines bond together, not the anamine. So it would break it. It would break a chunk and then just replace it completely. It would just cut a chunk and replace it. Chromosomal is pretty easy. This one is.
I want deletion.
Why?
Because this it's the B enzyme that gets the
B just gone. B is gone.
That's gone.
What about this one?
That was duplication because the B nucleotide gets
doubled. It's unnatural. There's 2. What's this one?
That one is inversion. It's opposite down.
Yeah, the order is like messed up. This one is
Translocation. They switch locations.
it's because some of the stuff in here, like E and F and D and JKL should have switched. We're done.